Thank you for joining the Jackson Laboratories presentation on Protect Your Research, Know Your B6 Mouse. My name is Peter Kelmanson, and I've been with Technical Information Services at the Jackson Laboratory for over 11 years. C57 Black 6, or B6 inbred mice, the most commonly used mice in biomedical research. This inbred strain is the most well characterized, the first to have its entire genome sequenced, and is the genetic background strain of choice for most targeted mutations and transgenics. The universal acceptance and demand for B6 mice has necessitated their production from multiple sources, introducing genetic and phenotypic variability that has very important consequences for accurately interpreting and repeating research results. So if we just did a quick exercise and just looking at these three mouse strains, if I were to ask you which of these two were genetically the most similar, A and B and C, or A and C, most of you would likely answer B and C, as they both look like C57 Black 6 mice. They're both black. However, it turns out that actually A and B are the most genetically similar. Strain A is a mutation that arose in the colony of C57 Black 6J that makes the mice albino, but it's a single gene mutation. While as the C57 Black 6J, which is picture B, and the C57 Black 6NJ mice, which is picture C, those two strains have been separated since 1951, and they differ in many different mutations that are involved in metabolism, neurobiology, immunology, vision, hearing, as well as behavior. And that's really the heart of what I want to talk to you about today, is really knowing what Black 6 substrain of mice you are using so that you can best interpret your data. And just as another example to drive that home, in that coat color is not actually a very good indicator of genetic background. These are all mouse strains that are all C57 Black 6J backgrounds. And not only do we have the albino ones, but we have revertance to agouti, beige, the dominant spotting phenotype. So all five of these strains are all C57 Black 6J backgrounds, but they all have a different coat color. So again, coat color is not a very good indicator of genetic background. Really, you would actually have to survey the actual genome really to determine which strains are most closely related. At the Jackson Laboratory, we do have two substrains of C57 Black 6. We have both the C57 Black 6J, which is our stock 000664, and the C57 Black 6NJ, which is our stock 005304. Both of those colonies are maintained at a high health status. They're very well characterized strains, some of the most published strains that you can find. We have extensive phenotypic data available, as well as consistent data reproducibility, which I will talk to you a little bit later when I get into it discussing our GSP program. Before we start to talk about different substrains of C57 Black 6, we really need to look at the origin of the C57 Black 6 inbred strain itself to help understand the genetics of that before we can talk about the genetics of substrains. So back in the early part of the 1900s and earlier, there was a lot of people who sort of raised mice. They were called mouse fanciers. They raised mice. They had different interesting coat colors or different phenotypes, and they would trade them amongst each other or sell them. And one of them, her name was Abby Lathrop, and she was in Massachusetts, and she provided mice to the Bussey Institute at Harvard University. And it was also about that time that geneticists really realized that mice were an ideal model organism for studying mammalian genetics. I mean, they're small, they're easy to maintain, they generally have really good reproductive performance, and their anatomy and physiology was very similar to that of humans. And then again, really at the beginning of the 20th century, Dr. William Castle started using mice from Abby Lathrop as well as a lot of other people, and he again was at the Bussey Institute in Harvard. And then shortly after that, one of his students, C.C. Little, who was the founder of the Jackson Laboratory, he began inbreeding those mouse stocks as a student of Dr. Castle. And really, of course, that is the initial inbreeding which led to a lot of the classical inbred strains that we have today. And in 1921, C.C. Little got some of Abby Lathrop's pet shop stock. In this case, it was a mouse strain that had a Waltzer phenotype. It's a mutation that sort of causes this spinning, dancing behavior. And when he mated female number 57 to two different males, he ended up getting black mice, and the other he got brown mice. And as he started to inbred, there was a mutation arose in the brown colony that led to a leaden or gray phenotype. And so as those mice were inbred, they were called C57, because it was female number 57, and then BL for black, BR for brown, and L for leaden. As simple as that. So the way inbred mouse strains are generated is by brother-sister mating every generation. So the progeny of your initial cross, which are the F1 hybrids, that generation you take a brother and sister, you breed them together to produce F2 mice. Again, you take a brother or sister and you mate them together to produce F3 mice, and then you would keep on doing that. And after 20 generations, you would generate an inbred mouse strain. And with inbred mice, all the individuals are virtually genetically identical, so you have a high degree of genetic homogeneity, and the mice are homozygous at virtually every locus. And that way, with that high degree of homogeneity, you're going to have a high degree of statistical reproducibility. So if you take a group of inbred mice and you give them one treatment, and you take another group of the same inbred strain and you give them a different treatment or no treatment, any of the differences you see are going to be due to the treatment, not due to underlying genetic differences. As well as if you then introduce mutations or transgenes, the mice that have the mutation are transgene as compared to mice that don't, because they're otherwise genetically identical. Any differences you see will be due to the presence of the transgene or the mutation, not again underlying genetic differences. That makes inbred mouse strains a very, very powerful tool for studying genetics. When you are working with inbred mouse strains, there are really two things that you need to make sure you're very well aware of. You need to understand the basic genetics and mutations and phenotypes of the inbred strain in general, and then as you see coming up, you also need to understand what substrain of that inbred strain you are using and what mutations and phenotypes might be specific to that substrain. That way you can best interpret your results correctly. And I'll give you an example. All C57 black 6 substrains carry a mutation in a gene called cadherin 23, known as the age-related hearing loss, or AHL mutation. Therefore, all C57 black 6 mice, as they get older, they will start to lose some of their hearing. And this, of course, is going to cause a complication interpreting data that you have that influences diseases or phenotypes that involve hearing and neurobiology. For example, phenotypic analysis of genes that are implicated in cognitive behavior, like fear conditioning in older mice, will require an auditory cue, which could be problematic then using C57 black 6 mice. And this mutation can infect lots of research areas, including autism, anxiety and stress disorders, addiction, and cardiovascular functions. So it's really critical that you understand the basic mutations and phenotypes of the inbred strains you are using. But then you also need to look at the substrains and realize that substrains can develop relatively quickly. And so one has a substrain when a colony is separated by 20 or more generations or phenotypic or genetic differences are discovered. And it's relatively quick to generate these substrains. So if you have a C57 black 6 parental colony and lab A gets those mice and breeds them for 10 generations, and lab B gets those same mice and breeds them for 20 generations, those two generations add up, and the mice held by lab A and lab B are actually 30 generations apart. So it can actually go a lot quicker than you expect. And C.C. Little, who founded the Jackson Laboratory and is also the one who created the C57 black 6 inbred strain, and since he was at the Jackson Laboratory, the C57 black 6J then is the parental colony. But between 1930 and 1970, C.C. Little sent many of those C57 black mice to researchers and institutions around the world because he truly believed that an important part of research is sharing resources to make that research more robust, and so he certainly did that. And then you can see that many different C57 black 6 substrains have been established around the world. And in 1951, some of the black 6J mice were sent to NIH, and they became the black 6N mice. And those are the two substrains I'm going to talk a lot about today because those are probably the most two commonly used substrains of C57 black 6 that are used in biomedical research. So in addition to the C57 black 6J substrain and the C57 black 6N substrain, the Jackson Laboratory actually has additional substrains that have been in various places. And what you'll find is everything after the 6 is going to be the substrain designation, and that's going to use laboratory codes. The J is the laboratory code for the Jackson Laboratory, N is the laboratory for the NIH. And then you can see that, for example, we also have the C57 black 6 HAJ, which went to a Dr. Hauschka and then came to the Jackson Laboratory. The BYJ had gone to Dr. Bailey and then the Jackson Laboratory. And then the EIJ went to Dr. Eicher and then back to the Jackson Laboratory. And then, of course, the J is at the end of all those because we are the lab that is currently maintaining the strain. And if you are interested in tracing laboratory codes, there is this link at the bottom of the page to the Institute for Laboratory Animal Research, or ILAR. You can look up any lab code or request a lab code of your own if you do need those. And I've mentioned a couple times now already that not all C57 Black 6 substrains are the same, and that they will differ genetically in a variety of ways, whether it be single nucleotide polymorphisms or SNPs. There can be insertions, deletions, also known as indels. There could be copy number variation, either due to duplications of regions of the chromosomes, and all of these would be more spontaneous mutations. And these genetic differences can translate into phenotypic differences between different C57 Black 6 substrains. They can affect a variety of phenotypes, including those in metabolism, and I'll give you some examples from neurobiology, immunology, and others as well. So here's an example of comparing how the C57 Black 6J and the C57 Black 6NJ mice respond to a high-fat diet, also known as diet-induced obesity, or DIO. In this case, the mice were fed a 60 kcal% high-fat diet starting at 6 weeks of age. And as you can see on the graph at the right, the C57 Black 6J mice gain more weight than the C57 Black 6NJ mice. But I'd like you to imagine a scenario where, let's say you had a mutation or transgene on a C57 Black 6 background, and you put them on a high-fat diet. And what if your data sort of came in between those two graphs? Well, if you were comparing your data to that for the Black 6J, if that's what you were using as controls, that would indicate that your mutation or transgene had reduced the amount of fat put on. But if you were using C57 Black 6NJ mice, you may then believe that your mutation or transgene increased the amount of fat. So depending on the control that you use, the way in which you interpret your data will differ. And that's why it's really critical to understand not only what strain you're using, but what substrain of C57 Black 6 you are using, so that you can properly interpret your data and share that with the world. Here's again another example from the same reference, comparing a glucose tolerance test between C57 Black 6J and the Black 6NJ, also on a high-fat diet. And glucose tolerance test really measures the ability of the mice to clear glucose from the blood. So they're given a bolus of glucose, and then you measure the blood glucose levels over time. And what you can see is that the Black 6J mice, even a couple weeks, as well as it gets more pronounced after 14 weeks on the high-fat diet, can't clear glucose as well as the Black 6NJ mice. And again, the same situation, if your data fell in between those, depending on what you chose as your controls, would determine how you interpret the data. And if you choose the wrong substrain of Black 6, you may incorrectly interpret that data. And one can also see neurological differences as well. When one looks at mice from the C57 Black 6J mice compared to the Black 6N from Charles River, those two strains are wild type for a gene called SNCA, or it's an alpha-synuclein protein. But when you look at the Black 6 mice from Harlan, those mice have had a spontaneous deletion of that gene. It doesn't necessarily give a visible phenotype, but the alpha-synuclein protein is implicated in a wide range of neurodegenerative diseases, and it's that primary structural component of the Lewy bodies that are found in the brains of Parkinson's disease patients. So that mutation doesn't necessarily give an obvious phenotype. However, if you are studying some sort of neurodegenerative disease, that may affect the data depending on your experiment on using which substrain you've used. So it's really important, again, to understand not just the strain you're using, but the substrain of C57 Black 6, so that you can either choose the right substrain for your experiments or at least interpret your data correctly. And here's another example of a behavior in which the Black 6 mice, which are known to have a preference for alcohol over other strains. So if you give them a choice of water with alcohol or water without, they'll prefer the water with alcohol. But you can see that the Black 6J mice actually will drink more, so they'll consume more alcohol, and they'll have a higher preference for it than the Black 6N mice, which again leads to a different phenotype, again with two different substrains. Again, depending on what controls you use in your experiment, you may interpret your data incorrectly depending on your strain. But also, there's another example of having two different substrains which have a different phenotype, which has some notable differences now in gene expression, as you can see between the genes at the graph in the lower right-hand corner, PLAC9 and the D14 ERTD4449E, which could potentially be responsible for those different phenotypes. So this different substrains not only is an issue to pay attention to, but can be potentially good experimental models for looking at those different phenotypes and determining the underlying genetic differences. And the last neurological one involves vision. In this case, it's a gene known as CRB1, which is localized to Muller cells and the photoreceptor inner segments. Mutations in that gene is associated with retinal diseases in people, including retinitis pigmentosa, as well as retinal degeneration. There's a mutation in that gene called RD8 for retinal degeneration 8 that results in this progressive spotty retinal degeneration in mice, which you can see in that picture in the lower right-hand corner. The left-hand retina is a normal retina, and the one on the right is the one with the RD8 mutation, and you can see that retinal degeneration there. All C57 black 6N substrains are homozygous for the RD8 mutation, but the C57 black 6J mice are wild type for that mutation. So that mutation happened after the mice had gone to NIH in 1951 and happened in that colony, and now all other derivatives or other C57 black 6N substrains are then homozygous for that mutation. And the consequences of that could be in the interpreting of genes or data where you're looking at either phenotypes that involve sight or involve neurobiology. Phenotypic analysis of genes that are implicated in cognitive function, like behavioral tests like the Morris-Water maze, which requires visual cues. If you have C57 black 6N mice, they're going to have vision problems, which means they're going to respond differently due to that mutation, not necessarily due to any treatment or other mutations or transgenes that you have. And this can impact areas of research like Alzheimer's or autism, Down syndrome, Rett syndrome, and a whole host of other neurodegenerative disorders. So it's really critical that you understand that if you are using those mice, because that could be an issue, again, in properly setting up your experiments and interpreting your data. And this has really come to be a sort of a major issue with the retinal degeneration in the black 6N mice, as there's this International Knockout Mouse Consortium, or IKMC, which is an international project to knock out all of the protein coding genes in the mouse. But it just so happens that the ES cell lines used to create all these mice were from a C57 black 6N substrain, meaning that all of these mice are homozygous for that RD8 mutation. And this, again, is a combination of strains from the Knockout Mouse Project in the US, the COMP, the European Conditional Mouse Mutagenesis Project, or EUCOM, and then also the NORCOM group in Canada. And then there's also the Texas A&M Institute for Genomic Medicine, TIGIM, which is a separate thing, but where they're making gene traps. But again, they're all in C57 black 6N substrains, which means C57 black 6N becomes the better control, but will have the retinal degeneration phenotype. So if you're looking at behavior that requires vision, it's going to be an issue. In addition to generating all of these knockout mice, the IKMC also has designated 3 different phenotyping centers to start collecting phenotypic information for all of these different mutations, and have looked at reproducibility. And they did look at comparing the black 6J and the black 6N, and looking at a whole series of data. As you can see here, this is just a subset. And the way this data is indicated is any of those rectangles that are in red indicates that the black 6N have a higher response than the black 6J, and the darkness of the red indicates then a higher difference. The rectangles in green indicate that the black 6N has a lower value than the black 6J. And again, the darkness of the green color indicates the strength of that decrease. And they also looked at males and females for both. And here you can see all this data that's comparing it, and it's relatively very well consistent between the 3 different phenotypic centers that are being used in that project. And some of these data that has been collected here is just an example of an immunological difference where the black 6J females actually show greater susceptibility to infection with Listeria than any of the black 6J males or the black 6N mice altogether. And then you can also see that the black 6N mice show a significant pro-inflammatory response 3 days after infection, where they have significantly higher expression levels of IL-6, IP-10, and CCL2. And another example out of this data set is the black 6J mice, looking at which have a higher response with delayed type hypersensitivity, which is a experimental protocol where you sensitize the mice by challenging them with DNFB on the stomach, and then you come back and challenge them with it on the ear. And what will happen is they'll react to that by ear swelling. So they'll get inflammation in the ear, and then you can just measure the ear swelling as a physical measurement, and it is a T cell mediated response. And what you see with this is that the Black6J males and females have a significantly greater inflammatory response than the Black6N males or females. And then also one can look at, there's a lot of information known about the genetic background in terms of mutations and SNPs of the Black6J and Black6N, and there multiple of those mutations have been identified, as well as structural variants. That data is available as well. And this knockout mouse phenotyping project, or the COMP2, is to generate and phenotype 2,500 different knockout mouse strains, and it's a collaborative effort between the 3 genotyping centers. The Jackson Laboratory here in Bar Harbor, Maine, is one of those centers, along with the Baylor College of Medicine in Houston, which also is working with the Wellcome Trust Sanger Institute and the MRC in Harwell. And the third place with the phenotyping center is the UC Davis, which in addition to their facility is working also with the Toronto Center for Phenogenomics in Canada, and the Children's Hospital Oakland Research Institute, and the Charles River Lab in Massachusetts. So when we look at the genetic background of the mice distributed by the Jackson Laboratory that are on a C57 Black 6 background, whether they're knockouts, transgenics, or spontaneous induced mutations, nearly 2,000 of those mice are on a C57 Black 6J background. We have about 70 that are on a Black 6N. However, we have over 1,000 strains either coming from the COMP2 project or through UCOM that are going to be on the C57 Black 6N background. And so understanding the differences between those backgrounds and understanding the nomenclature differences is going to help ensure that you can pick the right control strain for the strain that you're getting, and that you understand that there are differences between them. And so of course when you start looking then with these different substrains, we have to talk about the controls and how you choose the right control. So if you have a congenic mouse strain where you have a mutation or a transgenes that's been back-crossed to C57 Black 6, whether it's Black 6J or Black 6N, for more than 10 generations, you can typically use litter mates as controls if you're doing het by het or het by wild type, or if it's a transgene hemi by wild type, if any of those mating schemes. And in those cases, you can always use wild type or potentially the heterozygous litter mates as controls for the mutant gene or allele. If you have transgenic mice, you would use the non-carriers or the wild-type mice. But you could also potentially use non-litter mate controls from the colony. So that would be, say, mice from other litters that are all from the same colony. If you're breeding homozygote by homozygote, then you could use the inbred substrain as your control. But again, you need to make sure you understand what substrain your homozygous mutants are on so that you can match the background as close as possible to make sure you have the best possible control. If you have a mixed background, say a mix of Black 6J and Black 6N, which is actually more common than you might think, and I'll discuss that in a little bit coming up, but it's not uncommon for people to not either know what background their Black 6 mice are on, or potentially to get mice from one vendor one time, say Black 6J, but then the next time maybe somebody's got a little discount going on, so they get Black 6N the next time, and now they have a mix of both. And if they don't understand there are significant genetic differences between those, they may not be overly concerned. But in those cases, really your only options are litter mates. So that would either be wild type or heterozygous for the mutants, or non-carriers for the transgenes, or again your non-litter mate controls from the colony. I'd like to give one more example. So if you remember back when I was talking about a high-fat diet and the diet-induced obesity, I gave that hypothetical situation. I actually want to show you actual real data that has been published that ran into that same conundrum with choosing controls. So these are researchers, they had generated a JNK2, also known as MAP kinase 9 knockout on a C57 Black 6 background. And what they did is they then used acetaminophen-induced liver injury was their phenotype they were looking at. So they compared that knockout to the C57 Black 6J controls. And what they found is that the knockout mice had a higher degree of liver injury, indicating that the MAP kinase 9 gene product, or the protein, was protective of that phenotype, which turned out to be the opposite of what the researchers believed was going to happen with those knockout mice. So they then compared then the knockout to the C57 Black 6NJ, and they then found that the Black 6NJ had a higher amount of acetaminophen-induced liver injury than their knockout mice. So that left him in that situation where if you compare the knockout to the black 6J mice, that indicates that gene is hepatoprotective. But if you compare it to the black 6NJ, that indicates that the JNK2 protein is hepatotoxic. So that left him in a position where now, what is the best substrain for their experiments? Then they actually looked at the genome and they found that it turns out that their mouse was actually on a black 6N genetic background, not black 6J. Therefore, the black 6NJ is the better control, and it looks like then that protein is hepatotoxic. So it's that protein itself is involved in that increased acetaminophen-induced injury. But again, if they didn't choose the right control, or they didn't have done both, and they had just looked at the black 6J, all of their data would be reported incorrectly. So they would have interpreted that wrong, and then everybody else who may have followed that research and built on it would have been doing things incorrectly potentially. So if you are getting any new mouse strain, whether it's from a colleague or a collaborator, or somebody published a strain and they're willing to give you mice, there's a lot of information you really want to ask them so that you can understand the genetic background of the mice they're sending you, and so that way you can choose the right controls. So you want to know what strain was used to develop the strain. Was there an oocyte donor involved? What was that? Was it an ES cell line? What ES cell line was used? Any other strains that might have been introduced through breeding? Did they cross it to a Cree or a flip strain? Did they cross it to some sort of reporter, like a LAC-Z or a GFP reporter, or other mutations? Did they remove it, or are those mutations still there? What's the current breeding scheme? Are they breeding het by het, home by home? What are they doing? It's nice to know the current generation. Been cryopreserved at any point? If so, what generation? Has it been backcrossed to an inbred strain? If so, what inbred strain? Have they confirmed the genetic background? So if they say the mice have been backcrossed 10 times the C57 black 6J, have they actually confirmed that it really is well backcrossed? And so at the Jackson Laboratory, we do have stringent QC quality control that we then look at the genetic background of all the strains that we import. And when we do that, it's not uncommon that we find that the genetic background of the mouse that we import is actually not what we are told. In fact, almost I think it's 31% of the strains that we look at are not on the background that we're told. So that's about 1 out of 3. I think something like 16% end up being on a mix of black 6J and black 6N. And so if that's happening with the strains that we import, just think about what's going on with the mice that you might get from a colleague or a collaborator or some other researcher. So it's going to be the same. So if you are getting a new strain, highly recommend that you look at monitoring the genetic background. And at the Jackson Laboratory, we do have services that can help you with that. We do have a genome scanning service that can distinguish between the C57 black 6J and the C57 black 6N substrains. So 150 markers with markers distributed evenly throughout the genome. So we can distinguish those two. We can also distinguish whether the mice have been backcrossed, say, to C57 black 6, whether it was on a 129 or went from BALB-C. We have panels that we can use to distinguish between any two commonly used inbred mouse strains and confirm the background. And I would highly recommend, if you are getting a new strain from other people, I would highly recommend you take that time and look at confirming the genetic background, because it's better to know up front that the mice are not the background you expect them to be, so that you can then plan accordingly, whether alter your controls or potentially backcross. But it's better to know before than after you get a lot of data, potentially find the problem, or you may never know there's a problem, and there you end up with the wrong control, and again you would be in a position where you would interpret your data incorrectly. I mentioned earlier that the Jackson Laboratory, we do have 2 substrains of C57 black 6, both the black 6J and the black 6NJ. Again, these are very well characterized. We have them both at a high health status, and I had talk about data reproducibility. So, what are we doing here at the Jackson Laboratory to ensure the genetic integrity of these 2 substrains? And so what we have developed is a genetic stability program. This is a patented program that we developed here at the Jackson Laboratory that we use to diminish cumulative genetic drift and thereby stabilize the phenotype of those mice. And so what we have for these strains is a 25-year supply of frozen embryos from the same generation. And what we do is we thaw out some of those embryos to generate our foundation colony. And the foundation mice then breed for at the most up to 5 generations. After that point, we would remove them from the shelves and generate new mice from what we have cryopreserved. During the 5 generations of breeding the foundation, we then pull off mice for the expansion and distribution, where those mice breed at for most 10 generations, which would then, of course, be the mice that you would be purchasing directly from us. So therefore, the mice that you get from us for either the Black 6J or the Black 6NJ are at most 15 generations removed from what we have cryopreserved. Now, during those generations, mutations will happen, but because we keep going back to the beginning, those mutations are not able to fix or become homozygous in the colony. Therefore, the mice that you get from us today are going to be as genetically identical to mice that you get 5, 10, 20 years from now. Thereby, we're then maintaining that genetic stability and then retaining that same phenotype. So again, that the phenotype and the genotype should stay stable every year. And we have a lot of resources that we can use to support using Black 6 mice. We have the mouse phenome database, where we're collecting baseline phenotypic information for a host of commonly used inbred mouse strains, including the C57 Black 6J, where we have close to 3,000 different measurements. So you can use that to either get baseline information for the phenotypes you're interested in. That way you don't have to establish them yourself in your lab, but you just can validate them. There is whole genome sequence data available from the Sanger Institute and the Mouse Genomes Project. The C57 Black 6J was the original mouse genome that was originally sequenced, but the Black 6NJ has been sequenced with this Mouse Genomes Project. Therefore, you can search for mutations between those. We also have a lot of preconditioned mice for the strains where we, including streptozotocin-induced diabetes or diet-induced obese, where you can get mice, Black 6J mice that are already fed a high-fat diet, as well as aged mice as well. And then both of these strains are available at a very high health status with no additional charges for that. It's all the same. You know, I just want to make sure you guys are aware again, that's really critical to understand the phenotypes of the strains you're using, and you can use the mouse phenome database, or MPD, to get some of that baseline phenotype. And here's just an example of plasma glucose levels after a 4-hour fast, and looking at 43 different mouse strains. The arrow is pointing to the Black 6J. So you can use, of course, the MPD database if you're using any commonly used inbred strain, not just C57 Black 6. But if you are using Black 6, which most of you certainly are, you can find baseline data in that database, which you can then have an idea of what to expect prior to any treatment or mutation. And so really you need to really consider that your experimental model is one of your most important reagents. You know, really choose wisely and really understand the inbred strain you're using as well as the substrain. The more you know, the less problems you'll run into because you really understand that. Always really critical to use proper nomenclature. So if you're using Black 6 mice, indicate are you using Black 6J, are you using Black 6NJ, Black 6NCRL. It's really critical to really use that. That way anybody else who reads that publication will know what strain you used. And then again, you know, minimize genetic drift. Either get your mice from a respected vendor that is working to minimize genetic drift, or if you've got a mutation or transgene on a Black 6, say J inbred background, you want to back cross to the Black 6J every 5 to 10 generations to keep your mice as similar to your controls as possible. Make sure you're well educated and really establish a good quality control culture, because really the better you understand and the better quality control you take, then the less mice you'll have to use. And really, really good science does result in using reduced animals. So the less mice you have to use, the less resources and time are involved in that will improve your research program. So again, just to summarize, there are many different genetically and phenotypically unique substrains of C57 Black 6 that have developed over time, and they are going to continue to develop as time goes on. And really knowing and understanding the Black 6 substrain that you're working with is really critical to choosing the proper controls and proper data interpretation. Looking at the phenotypes between Black 6 substrains can also allow for identification of unique modifier alleles. So while you may think that the different substrains might be a headache to have to pay attention to, they may offer really, really good experimental models to look at those phenotypes that are different. And at the Jackson Laboratory, we're using our genetic stability program to limit genetic drift to stabilize those phenotypes over time. This presentation was made possible with your support. Jack's Mycin Services is dedicated to providing you with quality resources and flexible solutions and innovative technologies and unsurpassed expertise to support all aspects of your research. and realize that every dollar that you do spend at Jack's Mice and Services does support our shared research goals and sustains all these valuable mouse models and resources that are only available from the Jackson Laboratory. We do offer a host of services that might be helpful to you. Besides just having the specialty strains, we also do basic, custom, and complex breeding capabilities. So if you're having trouble breeding or you wish, we can breed Crelox mice together for you and either send you the breeding pairs or maintain cohorts for you here, as well as doing compound efficacy testing and genome scanning as well. And with that, again, thank you for attending this presentation. And if you do have any other questions, you can contact technical support at this link here, or you can contact your regional rep as well today if you want information from them, or schedule a visit if you'd like to get them in and talking to you directly.